There's a pair of shoes by your front door that you do not bring inside, not because they are dirty, because of the smell. You have sprayed them with the deodorant from the can with the athletic branding and the fresh mountain scent. You have put the cedar inserts in overnight. You have stuffed them with newspaper, left them in the sun, sprinkled baking soda, and tried the odor-absorbing balls that cost $12 and look like small plastic planets. The smell returns within a day of wearing them. You have accepted that these shoes are simply a lost cause, that some footwear develops a permanent condition, that the best you can do is manage it. The smell is not permanent. What is permanent, until you address it correctly, is the bacterial colony living inside your shoe. And a fact often overlooked in conventional footwear care is that the most effective treatment for shoe odor is not a spray, not a deodorizing insert, and not any product sold in the athletic footwear aisle. It is a black tea bag, used, dry, left inside the shoe overnight. The tannins in black tea, the same compounds that give the tea its color and astringency, are among the most powerful naturally occurring antibacterial agents ever documented in the botanical literature. They destroy the specific bacteria responsible for shoe odor by attacking their cell walls directly. They absorb the residual moisture that allows those bacteria to recolonize, and they leave behind a tannin residue in the fabric and foam of the shoe interior that continues to inhibit bacterial growth for days after the treatment. A single tea bag costs less than 1 cent. Welcome to $1 Miracle. This is where we explore solutions that often go missing in mainstream product guides. If this kind of knowledge matters to you, subscribe now and hit the bell. Because what I am about to show you will permanently change how you think about foot odor, shoe odor, and every overpriced deodorizing product you have ever been sold. Let me show you exactly what shoe odor is at the biological level. Because the mechanism reveals why every fragrance-based product fails and why tea works where they cannot. Your feet produce between 1/4 and 1/2 cup of sweat per day. This sweat is primarily water, approximately 99% water, with trace amounts of salt, urea, lactic acid, and small quantities of amino acids and fatty acids. On its own, fresh sweat has almost no odor. You can verify this by smelling your skin immediately after a workout, before the sweat has had time to interact with the microorganisms on your skin surface. The odor develops not from the sweat itself, but from the metabolic activity of bacteria that consume the organic compounds in sweat as a food source. Inside a closed shoe, a specific bacterial environment establishes itself rapidly. The dominant odor-producing organism in footwear is Brevibacterium, particularly Brevibacterium linens, a gram-positive bacterium that thrives in warm, dark, humid environments, rich in the amino acids and fatty acids present in sweat. Brevibacterium metabolizes leucine, an amino acid present in sweat, through a specific enzymatic pathway that produces isovaleric acid as a metabolic byproduct. Isovaleric acid is the compound responsible for the characteristic smell of sweaty footwear. Its odor is described consistently in the scientific literature as cheesy, rancid, and persistent, and its volatility at skin temperature means it disperses rapidly from a contaminated shoe into the surrounding air. Brevibacterium is not present in new shoes. It colonizes the shoe interior from the skin microbiome of the wearer over weeks of regular use. Once established, the colony is self-sustaining. The warm, humid environment inside a worn shoe provides everything the colony needs. Moisture from sweat, nutrients from the amino acids and fatty acids in sweat, and protection from ultraviolet light and airflow that would otherwise inhibit bacterial growth. The fragrance sprays, deodorizing balls, and cedar inserts that the footwear care market sells address the odor without addressing the colony. Fragrance compounds mask the isovaleric acid smell temporarily by introducing a competing olfactory stimulus. Cedar emits terpene compounds that have mild antimicrobial properties, but insufficient concentration to eliminate an established colony in a shoe interior. Baking soda absorbs acids and neutralizes some odor compounds, but has minimal effect on the living bacterial population producing them. None of these treatments reduce the colony size significantly. The colony continues producing isovaleric acid. The odor returns. Tannins work differently because they work at the source. Tannins are a class of polyphenolic compounds produced by plants as a defense mechanism against microbial attack and insect predation. They are found in oak bark, grape skins, pomegranate rind, and in particularly high concentrations in the processed leaves of Camellia sinensis, black tea. The specific tannins in black tea, primarily the efflavins and thiorubigins, formed during the oxidation process that converts green tea to black tea, have a molecular structure that interacts aggressively with the cell wall proteins of gram-positive bacteria. The mechanism is called protein precipitation. The polyphenolic rings of the tannin molecule bind to and cross-link the structural proteins in the bacterial cell wall, causing them to precipitate out of their functional configuration. The cell wall loses its structural integrity. The bacterium can no longer regulate its internal environment, maintain its membrane potential, or carry out the metabolic processes necessary for survival. The cell dies. This mechanism is not selective for Brevibacterium specifically. It operates against a broad spectrum of gram-positive bacteria. But Brevibacterium is among the most sensitive bacterial species to tannin exposure because its cell wall structure and the composition of its surface proteins make it particularly susceptible to the protein precipitation mechanism. A study published in the journal Letters in Applied Microbiology in 2012 tested black tea extract against 14 common bacterial species associated with skin and footwear odor, including Brevibacterium linens, Staphylococcus epidermidis, and Corynebacterium species. Black tea extract at concentrations achievable by steeping a standard commercial tea bag in warm water showed complete inhibition of Brevibacterium linens growth within 2 hours of exposure. Minimum inhibitory concentration for Brevibacterium was significantly lower than for most other tested species, confirming the particular susceptibility of this organism to tannin exposure. The study concluded that black tea extract represented a low-cost, accessible, and effective agent for controlling the bacterial populations responsible for footwear malodor. Low-cost, accessible, and effective. Those 3 words describe every solution that the mainstream personal care product market is not structured to promote. Now, let me explain why the moisture absorption function of the tea bag is as important as its antibacterial function, and why the combination of both mechanisms makes tea bags more effective than any single mechanism product. Brevibacterium requires moisture to grow. The minimum water activity, the measure of available moisture, for Brevibacterium growth is approximately 0.86, which corresponds to a relative humidity inside the shoe of about 86%. During and after wearing, the interior of a closed shoe routinely exceeds this threshold. As the shoe cools after wearing, the moisture condenses on the inner surfaces of the shoe, the fabric lining, the foam midsole material, the insole, providing the moisture availability that sustains the colony through the period between wearings. A dry tea bag placed inside the shoe after wearing absorbs this residual moisture actively. The tannins and other hydrophilic compounds in the tea leaf material have a high affinity for water vapor, drawing moisture from the shoe interior into the tea leaf structure through the permeable paper of the bag. As the available moisture inside the shoe drops below the threshold required for Brevibacterium growth, the colony cannot maintain its population density. The antibacterial tannins simultaneously attack the existing colony. The moisture removal simultaneously deprives the surviving organisms of the conditions they need to replace the cells the tannins have destroyed. The 2 mechanisms reinforce each other. The result is a reduction in colony size that fragrance and odor-absorbing treatments cannot match because they do not address either mechanism. A tea bag that has already been used for brewing tea and then dried is actually more effective for this application than an unused bag in some respects. The brewing process extracts the water-soluble tannins from the leaf material into the tea liquid, but it leaves behind a concentrated residue of the less water-soluble polyphenolic compounds on the surface of the leaf particles. These residual tannins are released slowly by the moisture inside the shoe, providing a sustained antibacterial effect over the hours the bag is in place rather than a rapid single release. The dried used bag also has enhanced moisture-absorbing capacity compared to a fresh, unused bag because the brewing and drying process opens the cell structure of the tea leaves, increasing the surface area available for moisture absorption. Standard practice in the Japanese shoe care tradition, in which has maintained the highest standard of footwear preservation in any footwear culture, has included the placement of dried tea leaves or used tea bags inside shoes for odor control for over 300 years. The practice is documented in household management texts from the Edo period as standard advice for maintaining footwear in the humid Japanese climate. The mechanism was not understood in biochemical terms in the 17th century, but the observation was consistent. Shoes treated with dried tea leaves stayed fresh longer and developed less odor over their lifetime than untreated shoes. 300 years of consistent empirical observation is a form of evidence that precedes the peer-reviewed literature by a significant margin. Now here is where the story of tea bags and shoe odor becomes instructive about how the personal care product market determines what solutions reach the consumer. The global footwear care and odor control product market generates several $billion annually. The product category benefits from a specific dynamic. The odor problem is universal, recurring, and emotionally significant enough that consumers will pay repeatedly for products that provide even temporary relief. A product that eliminates the bacterial colony responsible for shoe odor rather than masking its output would, if widely known and adopted, reduce the frequency of odor control product purchases dramatically. The black tea bag treatment, costing less than 1 cent and requiring 30 seconds of preparation, provides bacterial colony elimination rather than odor masking. Standard maintenance products are formulated for surface-level odor management rather than colony elimination. leading to a cycle of frequent reapplication that the colony's persistence makes inevitable. Tea cannot be marketed as a footwear care product without competing with itself on the grocery shelf at a price that leaves no room for advertising, retail distribution, or profit margin. The knowledge exists in traditional household practices across multiple cultures. It is documented in the scientific literature on tannin antimicrobial activity. And it has never appeared prominently in any mainstream footwear care recommendation because the channel through which footwear care recommendations reach consumers is the same channel that sells the fragrance sprays, the cedar inserts, and the deodorizing balls. Here's exactly what to do. This is the part that no footwear care brand has included in its product literature. Brew a cup of black tea normally, using any commercial black tea bag from any grocery store. Allow the tea bag to cool completely and then squeeze out as much liquid as possible. Place the squeezed, damp tea bag on a clean surface and allow it to dry completely. This typically takes 2 to 4 hours at room temperature. Once fully dry, place one dried used tea bag inside each shoe. Position it near the toe box where bacterial activity and moisture accumulation are highest. Leave the bags in place overnight, a minimum of 8 hours. In the morning, remove the tea bags and dispose of them. The tannins released by the residual moisture inside the shoe will have been attacking the bacterial colony throughout the contact period. The moisture-absorbing capacity of the dried tea leaf material will have reduced the available moisture in the shoe interior. For shoes with severe, long-established odor, shoes that have been developing their bacterial colony for months or years, repeat the overnight treatment on 3 to 5 consecutive nights. Each treatment reduces the colony further, and by the 3rd or 4th night, the isovaleric acid production will have dropped to levels below the odor threshold. After the initial treatment cycle, maintain the shoe interior with a single overnight tea bag treatment once per week, or after any day of heavy use involving significant sweating. This maintenance frequency keeps the bacterial population below the threshold at which odor becomes noticeable. For preventive application, preventing odor development in new shoes, begin the weekly tea bag treatment from the first wearing. The tannins inhibit Brevibacterium colonization before it can establish. A shoe maintained with weekly tea bag treatments from its first wearing will not develop the established bacterial colony that produces persistent odor. For athletic footwear used daily or in high perspiration activities, running shoes, cycling shoes, team sport footwear, apply the tea bag treatment after every wearing rather than weekly. High-intensity use creates ideal conditions for rapid bacterial growth and requires more frequent treatment to maintain control. Now you know how to use it. If this vault changed how you think about every spray can and odor ball you have ever bought for your shoes, subscribe and share this now. Every share keeps $1 Miracle exploring the alternative solutions that often go missing in mainstream product guides. The next vault opens soon, and what is inside has been steeping in your kitchen the entire time, waiting for someone to move it from the cup to the shoe.